Welcome to my world, the world of a medical laboratory scientist. Health is wealth, knowledge is power. Today's topic has to do with E. coli and early onset of colon cancer. Yes, majority of us must have heard of E. coli, Escherichia coli. It is a resident of the gut. You find it residing as normal flora. When we talk about normal flora, we are saying bacteria that do not cause problem. But in children, it has been discovered, especially children that are below 3 years or 4 years of age, it has been discovered that E. coli could cause diarrhea problem. And they have different strains, not all E. coli. We have the E. coli that will not cause any problem, but there are E. coli that will cause problem, and those ones have strains that have been grouped producing diarrhea. So the diarrhea in children, the normal thing we'll do is after we have cultured this stool of these children, we look out for E. coli and then try to find the strain that it belongs to, so that if it belongs to the diarrhea producing strain, then we can do something on getting the antibiotics that would really work and cure that infection. Though that is not a very difficult work for experts in the field. But these days, now we have a situation in our hands where it has now been discovered that there are some E. coli that produce the genotoxin. That is, they are called colibatin producing E. coli. They produce toxin in children around that age, and up to 10 years you start seeing the mark. They have deformed or transformed some of these DNAs to now be carcinogens, more or less, so that at a later stage you find people who are even under 55 years of age coming down with colon cancer. And that is where E. coli, colibatin producing E. coli, has been incriminated. And that is a study that when we go into the slide session, I'll be able to throw more light and grant more understanding on what the medical laboratory scientists, or what we, who are not even medically oriented, can do in order to reduce or stem the tide of colon cancers, especially the early onsets. So, as I used to say, press the like button, subscribe, and join me in the slide section. Outline: objective introduction, research findings, lab diagnosis, prevention, and recommendations are all I shall touch in this video. The objective: to create the necessary awareness of the colibatin genotoxic molecule produced by some E. coli strains and how it could be effectively diagnosed in the laboratory to stem the tide of early onset of colon cancers. The introduction: Escherichia coli, also called simply E. coli, belongs to the family of bacteria that resides in the colon that are collectively called coliforms before speciation. However, some E. coli strains have been incriminated in causing gastroenteritis, diarrhea, and enteric fever because they produce toxins that disturbs the microbiome homeostasis, the normal the intestine, apples, or various type of organisms that provides vitamins and what have you to stabilize that region. But at times there are toxins producing E. coli, and these are called the Shigella type, and these are what I have listed: invasive, enteropathogenic, entero. This table shows the kind of infection, common symptom, group most affected, major sources. Like I had earlier said, the children are most affected. For Shiga toxin E. coli, you have bloody urea symptoms, severe stomach cramp, vomiting, etc.
and the group most affected are children younger than 5 years. Adults 65 years and older and immunocompromised people are also affected. Next are the enterotoxigenic E. Coli, where the symptoms is watery diarrhea, stomach cramps, vomiting, and the children affected, younger adults, 18 years to 35. That is usually what you call international travelers, when you have this vomiting or discomfort after traveling. The source is food, water, or ice contaminated with animal poo or poo of an infected person. So when you talk of adults being affected, you are talking of this discomfort you experience when traveling. Yes, there is also enteropathogenic E. coli, the same experience, but you can see that the children affected are children that are younger than 1 year. Enteroinvasive E. coli, watery diarrhea is sometimes the symptoms, bloody, and children are mostly affected, international travelers. Yeah, enteroaggressive E. coli, this is also the picture. It's all children and people with living with HIV. They are already immunocompromised, and with the watery urea too, children usually 3 to 5 years of age. Some of these E. coli can cause some other symptoms, especially when they are disseminated by enteric fever through the blood to other organs like the kidney, etc. Colibactin on its own is a genotoxic molecule produced by certain strain of E. coli, particularly those with PKS gene island. It is a peptide polypeptide hybrid that can induce DNA damage, including double-strand breaks and chromosome aberrations. It promotes colon tumorigenesis and is associated with colorectal cancer risk, according to a study on Science Direct. So it can also cause cell circular arrest and senescence of epithelial cells. So these this new strain, identified strain of E. coli, could do serious damages to the DNA. That's why they call it genotoxic molecule, that it produces the toxin. Research findings. In a study, Alexandro and his colleague examined cancer genomes from patients with both early and late onset colorectal cancer across 11 countries. That is a very wide study. So comparing those who have early onset, people who are under 55 years, and the late onset under above 70, the team found that this colibactin producing E. coli leaves behind a distinct DNA fingerprint on colon cells when kids are exposed to it. These mutations were 3.3 times more common in early onset cases than in those diagnosed after the age of 70. Colibactin-linked mutations show up at the early stage of tumor development, they found, often reaching the first 10 years of life. Laboratory diagnosis. The laboratory investigations could make use of these quotes in early diagnosis. This is because we've not been checking for colibactin producing E. coli. So it's a new investigation. The PKS gene island is found in some E. coli strains, particularly those in the B2 phylogroup, which are often extraintestinal pathogenic. This island encode the biosynthetic pathway for colibactin. So it may be a need to do searching that will involve a routine check of this B2 phylogroup. And 2, colibactin leaves behind a distinct DNA fingerprint on colon cells when kids are exposed to it. So the fingerprints could be discovered using the NA8, that is nucleic acid application, and the PCR DNA test could also be done. The recommendation, apart from laboratory tests being improved to the standard of being able to identify these colibactin producing E. coli. Children who experience gastroenteritis early in life should be allowed to undergo a nucleic acid amplification test to detect the specific colibactin pKa sequence that is indicative of the infection by colibactin producing E. coli. The checking of the sample for random checking to know whether there are strange E. coli apart from the known ones, so that further tests could be done on these strange E. coli is also to be encouraged. The investigation for the colibactin producing E. coli is not yet a routine, but it is necessary for one to look at how we can apply this life-saving information. We could even perform sensitivity testing on any stool culture that is predominantly E. coli, even when they are non-strain compliant, that is, they don't belong to any known strain. Yes, we could also embark on tests on the children that are over 10 years old to look for that fingerprint, the mark that will indicate that, yes, they carry these genetic mutants. And many other things could be done. But for now, it's just thinking of how we can put to effect this life-saving information and help to reduce early onset of the colorectal cancer. If you like my presentation, please click the like button, subscribe. This is the 178th video that I'm producing, passing on information that we are very good.